Biological macromolecules are large organic molecules. Okay, so we finished up with the inorganics. Now we're moving on to organics. So these big molecules, remember macro means big, always contain carbon, hydrogen, and oxygen. There are some of our very important macromolecules that also have nitrogen, phosphorus, and sulfur, or any combination of them. But the one thing they all have in common, carbon, hydrogens, and oxygens. A single carbon atom or a carbon skeleton, which is a bunch of carbons all hooked together, can have only hydrogen atoms attached. Okay? They contain carbon bound to carbon or carbon bound to hydrogen. Okay, so those are hydrocarbons. Biological macromolecules are not just hydrocarbons though, because they also contain something called a functional group. Functional group is when you have two or more atoms that when they're together and present on a molecule, they always have the same chemical characteristics. So functional groups are polar and they form hydrogen bonds. And that increases the molecule's solubility in water, which means it can dissolve or dissociate, right? Some functional groups act as an acid and release hydrogen ions, such as carboxyl groups. Others may act as a base by binding to those hydrogen ions, such as the amine group. Biological macromolecules typically have more than one functional group within them, and we have 4 major macromolecules we're going to discuss. Oh wait, before we move on, I need to back up. This 4th bullet right here, polymers are made of monomers. Again, I'm going to make you think about your Legos that you played with when you were a kid. Each individual Lego by itself is a monomer. Mono means one. Mer means piece or unit. But if we hook a bunch of Legos together, then we no longer have a monomer, we have a polymer, okay, polymer. So poly, many, mer, units. So down here we've got a Lego castle with Lego people and Lego horses. Those are polymers. Up here, each individual Lego is a monomer. So as we start learning the macromolecules, that's something to keep in mind. What is the polymer, but what are the monomers that make it up? Now remember back, we talked a little bit about this earlier. These big macromolecules are made through an anabolic process. Remember, anabolism, to build up. So we build macromolecules from the monomers. As you see here in this first little image, we've got 2 monomers, and if we remove this water, then we form a chemical bond between them. So this is actually a dimer. Di means 2, okay? So if we continue to do this and make a big long chain of these little green hexagons, then we would have a polymer, okay? Now, hydrolysis is a catabolic process. Catabolic, remember, means to break down. So if we take this same little dimer that we made here and we add water to that, it's going to break that chemical bond, and now we're back to monomers. So just to give you an example of this, your book uses the same one I typically use in my face-to-face classes. Think of a polymer like a pearl necklace. The necklace itself, all the pearls may be absolutely identical to each other, but they are still individual pearls. So each pearl would be a monomer. The necklace would be the individual monomers hooked together to make a pearl necklace. That would be your polymer. Our first category of macromolecules are lipids. Lipids are a very diverse group of fatty, water insoluble, remember that means hydrophobic, so they're hydrophobic molecules that function as an energy storage. They help to make cell membranes and certain of our hormones. Triglycerides are the most common type of the lipids in living animals or in living beings. We are animals, by the way. They're used for long-term energy storage because we can make more adipose tissue. So adipose tissue is fat tissue, and we store those extra lipids in adipose tissue by forming triglycerides in a dehydration synthesis called lipogenesis. So when you hear genesis in A&P, I want you to think of making something or the beginning of. So we take extra lipids, hook them all together, make triglycerides, and we store them in adipose tissue when we have extra lipids in our diet. We're eating a lot of fatty, yucky foods, right? Now, because that adipose tissue, fat tissue, stores these triglycerides, we can also release them into the blood when nutrients are needed. That is a hydrolysis reaction that we call lipolysis. So the lipo part refers to lipid. Lysis means to break or split. Phospholipids are lipids with a phosphate group attached. Okay, so these are the main component of all of our cell membranes. Other than the fact that these phospholipids make up our cell membranes, another important thing to remember is that phospholipids are lipids with a phosphate group attached. The phosphate forms the head of the cell membrane, right? So the phospholipid with the phosphate heads, the heads are polar, so they are water loving, and they are oriented toward the intracellular and extracellular fluids. The hydrophilic fatty acid hydrocarbon tails, of course, those are going to be hydrophobic oils, hydrophobic, and they're going to orient away from the water. Steroids have their carbons arranged in rings, okay? Now, we're not going to get much into the chemical structure here. I'm just pointing out that they're very different from the long chains that we see in the triglycerides and the phospholipids. So your steroids are a type of lipids. They include cholesterol, which is a component of our cell membranes and gives it a little bit more rigidity, so they're not quite as flexible as they would be without the cholesterol. The steroids also are the fats that are a part of our steroid hormones, such as estrogen and testosterone. And also, steroids are a component of the bile salts that the liver makes and the gallbladder stores and releases into the digestive tract. The eicosanoids are the last class of our lipids that we'll examine. So you can see their structure is a little bit different as well. Now, guys, I'm not going to test you on the structure. I do want you to know that lipids are made of fatty acids and glycerol for the most part, okay? But you have those carbons, hydrogens, and oxygens, just like we've seen. So there are 4 classes of eicosanoids. They include the prostaglandins, prostacyclins, thromboxane, and leukotrienes. A lot of these are going to be very important signaling molecules that work with your inflammatory response when the immune system is activated, but they also play a role within communication of the cells within the nervous system. All right, guys, you just got your first taste of how things are with me in the classroom. If something's on the slide, man, I'm going to talk about it. So I just remembered the next 4 slides actually break down each one of these that we just did. So I'm not going to re-record on this video what we've already talked about. But these slides, triglycerides, phospholipids, steroids, and eicosanoids are the ones that we just covered on the slide that had all 4 of them in the one table. I'm sure you've all heard these terms before: saturated, unsaturated, monounsaturated, trans fats. All of this is referring to the chemical structure of these lipids. So most animal fats are what we call saturated. Most vegetable fats are usually, with a couple of exceptions, unsaturated. Most of the time you can distinguish between saturated and unsaturated by whether or not it's a solid or a liquid at room temperature. So most saturated fats are solid at room temperature. Most unsaturated fats are liquid at room temperature. We mentioned this before as well. Remember, with your saturated fats, all of your carbons are bound to hydrogens sharing electrons. In your unsaturated fats, a couple of these carbons don't have as many hydrogens attached, so they form a double bond and share electrons right here. Now, of course, this is going to not allow those carbons to be in a nice long chain. Instead, it's going to put a little bit of a kink in the chain, and therefore your unsaturated fats can't pile up. So if you think about this saturated fat, if you had another saturated fat above it and one below it, you could just keep on and build a whole wall of saturated fats. It'd be a little bit more difficult to do if your chain is bent. You can't pile them up. Now, unsaturated fats, though, can be converted to saturated fats by a process that we call hydrogenation, which increases the amount of hydrogen in the molecule. For example, vegetable oil can be hydrogenated to form margarine. Vegetable oil is a liquid, margarine is a solid, okay? So studies have shown, though, that partial hydrogenation of fatty acids can lead to something called trans fats. Trans fats in your diet raise the level of your LDLs, which are your bad fats in your body. It actually stands for low density lipoprotein. So the higher your LDLs, the higher your risk for developing buildup inside your blood vessels of these fats. That's something we call atherosclerosis and can lead to heart attack and or stroke. The term carbohydrate means hydrated carbon. So the carbons, or nearly every carbon in carbohydrates, are completely hydrated with what is equivalent to a water molecule. So you've got a hydrogen and an OH. Okay, put that together, what do you have? Water. The least complex of the carbohydrates are what we call monosaccharides, which simply means it's one sugar, one monomer. A disaccharide is when you have two sugars, monosaccharides, put together. Polysaccharides are formed from many monosaccharides. So let's go back to our terminology. The polymer is the carbohydrate. The monomer is the single sugar, which is called a monosaccharide. Here you can see some examples of monosaccharides. Some of them have 6 carbons, some of them have 5. Now, when you see these shapes like this, you're not seeing all of the carbons. Everywhere 2 lines come together to form a point, a carbon is there. We just don't draw them in. It gets a little messy and a little complicated. But look at all these H's and O's attached. So there's your water, right? HHO, H2O. Now, this is 5 carbon sugars, or 6 carbon sugars. These are 5. Now, I'm not going to ask you which is which. I'm just showing you some examples of these. But all of these are individual monosaccharides in the top box. Down here, we have disaccharides that are formed when we create a chemical bond between 2 monosaccharides. So if you look up here, the red is the fructose, the green is the ribose. There we have those 2, and oops, I'm sorry, that's a glucose. So we hook it together and we make sucrose. Over here, we hook 2 monosaccharides together to make lactose and maltose. So disaccharide, 2 sugars. Monosaccharide, 1 sugar. Polysaccharide, many sugars. All right, so glucose, as you see right here, just this, is a 6 carbon monomer. It is our most common and most important, in my humble opinion, most important monosaccharide because it is human gold. It is the primary nutrient that our body makes the most energy from. So think of glucose as gold, okay? Now, because of that, we have to very carefully maintain glucose levels in the body. I mean, you sort of need to maintain your gold or money level in your bank account to make sure you can do everything that you need to do, pay your bills, have a home, buy your groceries. Well, we need to make sure we have enough glucose for our body to do its job. So think of glucose as body gold. Just like you don't want to run out of money, our body doesn't want to run out of glucose. So what do we do when we have extra money? We put it in the bank so we can get it when we need it, right? Well, we do the same thing as humans. Immediately after we eat a meal, excess glucose is going to be stored primarily in the liver, but some in our skeletal muscle as well. So the liver and the skeletal muscle are really good at storing that glucose. The liver's going to take that glucose and hook it together and make a long chain branched carbohydrate out of all those individual glucose molecules. That carbohydrate is called glycogen. The liver stores it so we can release it when we need it. So glycogenesis is taking excess glucose and creating glycogen. Now, when our blood sugar levels drop, let's say you didn't get to eat breakfast this morning, you had to get up and go to school or go to work. By lunchtime, your blood sugar levels have dropped. So what's going to happen? Your liver is going to break down all that glycogen in a process called glycogenolysis. Remember, lysis means to break or split. So when the liver breaks down all of these chemical bonds that you see over here, holding all of these glucose molecules together, now we're back to individual little glucose molecules that we can release into the blood, and blood sugar level rises. Now, the liver has another pretty important job as well to help us regulate blood sugar, and that is that the liver can convert non-carbohydrates like proteins and lipids into being able to form glucose, and that's called gluconeogenesis. Neo means new, so creating new glucose. Now, all of this works together to make sure that our blood glucose levels, blood sugar, stay at homeostatic levels. And when it doesn't happen properly, we can develop conditions such as diabetes. Nucleic acids are the third type of macromolecule we'll look at, and their job is to store and transfer genetic information. We have two major classes of nucleic acids. You've heard of these, or at least I know you've heard of one of them. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Now, each of these are polymers, many units, composed of monomers called nucleotide. Those nucleotide monomers have three components. We have a sugar, a phosphate group, and a nitrogenous base. Okay, so we have four different nitrogenous bases. This one you see has a double nitrogen ring. Some of them have a single nitrogen ring. We have 5 types of these nitrogen-containing bases. And remember, these are nucleotides. We have 4 that are found in DNA and 4 that are found in RNA. So we have cytosine, thymine, adenine, and guanine found in DNA. In RNA, we substitute uracil instead of thiamine. So these 4 right here, cytosine, uracil, adenine, and guanine, these are found in RNA. In DNA, no uracil, we have thiamine instead. DNA is a double-strand nucleic acid that is arranged in a double helix or twisted form. Now, each of the strands has those nucleic acids, the A's, the T's, the C's, and the G's. Your DNA is located in chromosomes that are found in the nucleus of your cells and also within mitochondria. Now you're going to learn much more about these as we continue. So when you think about that structure of the nucleotide, you've got the deoxyribose sugar, the phosphate, and one of those 4 nucleic acids. So if you look over here at this image, you can see we've got A's, T's, C's, and G's. So no uracil, remember that's the one that's only found in RNA. So we've got 2 strands of A's, T's, C's, and G's. These little dotted lines between them are hydrogen bonds. What do you remember about hydrogen bonds? They're weak and they can easily be broken apart. Ooh, that's going to be important pretty soon. But now when we have these 2 strands of A's, T's, C's, and G's, they don't just randomly form hydrogen bonds between each other. Your A's, adenine, will always form a hydrogen bond with T, thymine. So A and T always go together, and your cytosine will always form hydrogen bonds with guanine. A goes with T, C goes with G, in DNA. Within this long strands of DNA that we have, there are smaller little pieces of DNA that we call genes. I want you to think of a gene like a recipe. Okay, if you want to know how to make beef Wellington, you open up a cookbook, go to the recipe, and it tells you what to put together and how to do it. Well, when our body cells need to make a protein, we open up a gene, look at the recipe, and it tells our body how to put those amino acids together to make proteins. You're going to hear about that one in just a second. Ribonucleic acid, or RNA, is sort of like DNA, but it's sort of different. So instead of a double strand, it's a single strand. Instead of T's, we have U's. So A and U, C and G. No thymine. Remember, we mentioned this right at the beginning of this one, okay? So the nucleic acids, DNA and RNA, have several differences, and we're going to continue to talk about this. But number 1 difference, single strand, DNA is double strand. The uracil in RNA replaces thymine in DNA. The sugars are also different. Instead of ribonucleic acid, we have deoxyribonucleic acid. Ribose is the sugar in RNA. Deoxyribose is the sugar in DNA. Remember, DNA stores our genetic information. RNA helps us get that information to where we need to go within the cell to turn it into a protein. So RNA carries the information so that we can use it. DNA stores the information so we can access it. Now guys, that's very simplistic. We're going to get further into this as we continue. Our 3rd nucleic acid is adenosine triphosphate, ATP. If I had $1 for every time I say ATP, I could buy the school. I'd live on the beach. I'd travel the world. Yeah, I talk about ATP a lot, but it's very important. ATP is our energy source. It's how we power metabolic reactions. Think about your car. You have to put fuel in your car for it to do its job. That fuel is gasoline. ATP is human gasoline. So it's a nucleotide that is composed of an adenine, which is a nitrogen-containing base, a sugar, and 1, 2, 3 phosphate groups. Tri means 3. So adenosine triphosphate. Okay? This is the molecule in the transfer of chemical energy that allows us to live. There are covalent bonds between the last 2 phosphate groups that are very unique and rich in energy, and they release that energy when we chemically break those bonds. Our last category of macromolecules are proteins. It's estimated that the human body makes about 50,000 different proteins. Yeah, isn't that absolutely crazy? And those proteins make up about 1/5 of your body weight. We'll look at some examples of proteins. So proteins have a lot of functions. They can cause synthesis or digestion. Synthesis means to make. Digest means to break. Okay? So proteins called enzymes are responsible for making and breaking. Structural proteins such as collagen give structure to body areas, to body organs, to body tissues. For instance, as we age, we lose collagen in our skin, and that's what causes wrinkles, one of the causes for wrinkles. We have membrane proteins that can serve as carriers to allow things to get across the cell membrane, either in or out. We have proteins that work as protection. Keratin is a protein found in our skin, also found in other areas, but it helps to provide some structure, but also helps to keep us from losing or gaining excess water. We have other protective proteins that work as antibodies with the immune system. Proteins like actin and myosin are found in muscles and allow for movement to occur. And then we have proteins that are transport proteins. They are found in the blood and they bind to things like lipids and carry them. And hemoglobin is a protein that carries oxygen from our lungs out to body tissues. A protein is a polymer, many pieces, many units. The individual monomers are amino acids. We have 20 different amino acids, but now think about that. I mentioned a while ago that we make around 50,000 different proteins out of 20 amino acids. So the number and the arrangement of these amino acids is what makes one protein different than another. For instance, if I told you my office number is 1234, well, if you dialed 4321, you're not going to reach me. It's different. It's made up of the same pieces, 1, 2, 3, and 4, but you put them in a different order, so it's not going to get the same result. Same thing with our proteins. They differ in the way the amino acids are put together. Now, your amino acids have an amine group, a carboxyl group, and something called the R group. So the amine and the carboxyl group are the same. The R group is what changes and makes each amino acid different from the others. So the amine group and the carboxyl, those are found in all amino acids. The R group is what is different. So when we take two amino acids and hook them together, we create a peptide bond, and this creates a dipeptide. Do you remember what di means? Oh, I heard all of you say two. Of course. Now, when we make a protein, we have multiple amino acids hooked together. Now, if it is a short strand of amino acids, anywhere from 3 to 20, we can sometimes refer to that as an oligopeptide. Oligo means short, but a polypeptide is long chains of amino acids or many oligopeptides all hooked together. Proteins are vitally important, and they're created in a very specific way according to the information in our DNA. Remember, I mentioned DNA tells our cells what proteins to make? Well, the sequence of A's, T's, C's, and G's in the DNA determines what order we put these amino acids together. Now, once the amino acids are put together, they change a little bit and interact with each other. So some amino acids may have a positive charge, others may have a negative charge. Now remember, positives and negatives attract. So let's say that this little blue amino acid right here has a positive charge, and this green amino acid right here has a negative charge. Well, that's going to cause this blue and this green, positive and negatively charged amino acid, to come together because opposite charges attract. That's going to cause the protein to fold. Now let's go down here. Let's say this blue amino acid is positively charged, and this blue amino acid is positively charged. Well, what's that going to do? If you try to put two positive ends of a magnet together, what do? They push away from each other. Well, the same thing's going to happen because of the charges on these two amino acids. Positives and positives will push away from each other. Negatives and negatives will push away from each other. But a positive and a negative will come together. And that causes the protein to fold. Now, there's much more that we're going to see in just a second. But if we alter the way that protein is supposed to be folded, it alters the function of that protein because it can no longer do its job. So that sequence of amino acids and the way that we put them together to be 1, 2, 3, 4 instead of 4, 3, 2, 1, that sequence is the primary structure of amino acids. So just how do we put those amino acids together to form a long chain of amino acids? The secondary structure is due to interactions that form hydrogen bonds between amino acids. So this is going to cause the long chain of amino acids to change into one of two shapes. A spiral coil that we call an alpha helix. We find a lot of these in structures that need a little bit of elasticity to them, such as hair and skin, or folded up into what we call a beta pleated sheet. I think of this like back when you were in elementary school and you took a piece of paper and you folded it one way, then the other, then one way, then the other, and just that back and forth folding to create a little fan that you could use while you're sitting in the classroom. That's pleated. So amino acids can form hydrogen bonds that cause that to happen as well. This is going to give flexibility. Now, elasticity allows stretch. Flexibility allows for bend. So a lot of our globular proteins, such as enzymes, will form the beta pleated sheets instead. Now, once we've got that, our third or tertiary level of structure, that is called, sorry guys, my dog was wanting out and I got distracted. Let me start this slide over, okay? Your tertiary structure is your third level of structure. So it's the final three-dimensional shape of a protein chain. We have two categories of these, and they're distinguished by their molecular shape. So globular, think of a glob of something. So globular proteins fold into a compact, almost spherical shape. These tend to be enzymes, antibodies, and some of the hormones. Your fibrous proteins are extended linear molecules like we find in the cytoskeleton, and proteins like actin and myosin, which are contractile proteins found within muscle. And our fourth and final level of protein structure is the quaternary structure. This only happens when you have two or more amino acid chains, okay? The classic example of this in the human body is hemoglobin. So in hemoglobin, we have two alpha chains and two beta chains. So a total of 4 strands of amino acids that get hydrogen bonds in their secondary structure. They fold up into their third level, tertiary level, and then if we have 2 or more of those that interact together, that provides a quaternary structure. So the 2 alpha helix or 2 alpha chains and 2 beta chains all come together. Each one of them bonds to a molecule of iron. That iron, of course, is going to bind to the oxygen, and this is what we have in our red blood cells that carry oxygen out to body tissues. As I mentioned before, proteins can be denatured. Denature, away from nature, which means that protein can't do that protein's job anymore. So it can cause it to be a little bit different, a whole lot different, excuse me, or perhaps it really didn't affect its function at all. But denaturation is usually irreversible. You can't put the egg back in the shell and make a baby chicken. So remember, proteins are created by chemical bonds being formed between amino acids. So if we disrupt those bonds by applying heat, by putting an acid in and altering the pH, mechanical agitation, think about just shake something until it falls apart, or if we add in other ions that can interfere with those chemical bonds, then the protein may lose its function. So if you look at this image, let's say the normal protein is folded like this. We add an acid or heat to it, break chemical bonds within the chains. Now the protein no longer looks like it does, so it can't fit or do the job it is supposed to do. That protein has lost its ability to function. And one last slide here for chapter 2. This is your overview of the major biological macromolecules, your lipids, carbohydrates, nucleic acids, and proteins. Be sure and take a look at this. You have these in your textbook. So when you see these big picture things like this, it is a direct copy from the concept overviews that you have in your textbook. This one is on page 62, 63. So you can get a bigger picture of this if you would like to look at it. And I always encourage you to look at this because it really does help you to tie everything together. I'll see you next in chapter 3.